Namaste. Welcome to Lecture 2 of Ayurveda, the Science of Life, an overview of Ayurveda from ancient to modern times. I'm Dr. Maya Murdeshwar from the Department of Life Science and Biochemistry at St. Xavier's College, Mumbai. In our first lecture on Ayurveda, we began with a discussion of the history of Ayurveda through stories and mythology, as also through the ancient practitioners, the sages, the Ayurvedacharyas, and the books or the ancient Ayurvedic texts that they wrote. We discussed the three dosha theory, the fundamental principle of Ayurveda, along with the dosha, dhatu, mala, and agni concept. We spoke about the Pancha Mahabhutas and how they make up the living systems. And we also discussed several Ayurvedic diagnostic and treatment strategies. In lecture 2, I will walk you through the history of Ayurveda once again, but this time through specific examples for the science and the scientific knowledge that is present in Ayurvedic texts. We will pick up examples from embryology, pharmacology, surgery, cardiology, and other branches of current modern medicine and look at how this science was mentioned in our ancient text 3000 years ago. We will then move on to comparing the approach of Ayurveda with that of modern medicine. We will look at how these are different and how they can be combined to an integrative approach to health and wellness. I will also discuss certain Government of India initiatives that have been set up by the Ministry of AYUSH to promote Ayurveda and other traditional medicinal forms in India. And we will conclude this lecture by discussing recent research in Ayurveda in the post-genomic times by referring to Ayurgenomics, network pharmacology, and P4 medicine. As we discuss all these topics, we will also look at the way ahead for Ayurveda. Let us begin now by discussing certain examples of scientific knowledge that is present in our Ayurvedic texts. In lecture 1, we looked at the definition of health and well-being as given in the Sushruta Samhita. Samadosha, Samagni, everything in balance in the body. The Charaka Samhita in its chapter 6, paragraph 13, also indicates the causative factors for this well-being. It is not just the balance of dosha, but what else is it that maintains health? So it states that the person has to be born in the place of strong men, in times of strong men, in a pleasant climate, with a healthy seed and uterus, should get nourishing food, should have a good physique, good maintenance of health, healthy mind, a natural disposition, youth, action, and cheerfulness. It further outlines that the causative agents for health and well-being are genetic, they are environmental. The environment also affects our health. The physiological causes, that is the physical causes, the biochemistry that happens inside the body, and also psychological factors, that is mental health, mental well-being. So a complete picture of wellness is mentioned in all the ancient texts of Ayurveda. It's not just health, but also disease that is spoken about. If you recall, the Roga Pariksha that was mentioned in lecture 1. So diseases are classified into three main types in Ayurveda: endogenous, exogenous, and cosmic. They fall under 7 subcategories, and these are the diseases that are caused because of preconception hereditary factors, genetic causes before conception. It is not just about health, but also about disease. Diseases in Ayurveda are classified into three major types: those that are caused by endogenous or inherent factors, those that are caused by exogenous or external factors, and those that have a cosmic or an unknown origin. They fall under 7 subclasses: those that arise because of preconception hereditary factors, genetic factors affecting preconception, that is in the parents; post-conception hereditary factors, genetic mutations that occur after the embryo is formed, the egg is fertilized by the sperm; a vitiation of humors or the aggravation of the three doshas; those diseases which are contagious, obtained through contact with external agents with other people; those which are seasonal, occur only in one particular season but not the other; those which are caused due to divine forces, which essentially means of unknown origin, unknown as yet; and those that are natural to the body. It's not just the classification, but also the evolution of diseases that is spoken about in the Sushruta Samhita. So I'll walk you through this slide where it talks about the evolution of diseases because of sanchaya, that is the accumulation of doshas; prakopa, the aggravation of those doshas; prasara, the dissemination of those doshas to various parts of the body. So it refers to systemic illness. Sthana sanshraya, localization of those doshas. So diseases affecting just a particular part of the body and not the entire system. Vyadhi, the actual manifestation of the disease. Bheda, the differentiation of the disease. And upadrava, that is the complications that result as the disease progresses. So a complete treatment of disease classification and evolution is found in Sushruta Samhita. Talking about the development of the human system, under embryology, all the three major treatises of Ayurveda, the Bruhat Trayi, describe the Masano-Masika Garbha Vruddhihi, which means the month-wise development of the fetus, right from the first month to the 9th month. What are the changes that are observed? Which organs grow during what time? Which systems are in place? These texts talk about the union of the shukra or the sperm with the shonita or the ovum, along with the atma in the garbhashaya, which is another term for the uterus. And this union is termed, the result of this union is termed as the garbha or the embryo or the fetus. This embryo is formed from the Pancha Mahabhutas according to the principles of Ayurveda, and the addition of Chetana or the soul to it gives life to this fetus. Garbha Vruddhi Kramaha, the sequence of development of events of the fetus, has been vividly described in all three texts. And it's not just the monthly development of the fetus, but cellular events right from this union of the sperm and the ovum to how that single cell, the embryo, develops, grows and divides and develops into this entire organism has been mentioned. Events which are not seen by the naked eye have been described over 3,000 years ago without the use of any modern day microscopes or cutting edge scientific instruments. Not just the growth and development, but the abnormalities, the developmental defects, birth defects that could possibly arise in this time have also been mentioned in these ancient texts. The ancient texts also talk about blood circulation in the fetus. In his compendium, which is called the Bhela Samhita, Maharshi Bhela talks about how or he asks the question, does the fetus in the mother's womb eat or not? So he says it does not eat, because if it would have eaten, there would be the creation of fecal matter. Then how does it exist inside the womb in a closed space for 9 months and grow and divide actively? So he says that in the navel of the child, a tubular vessel is present, is fixed, and this tubular vessel is connected to that of the mother. And through this tubular vessel, the mother satisfies and grows the fetus. So 3000 years ago, the placenta, the amniotic sac, the umbilical cord, and the blood circulation across, nutrients coming from the mother's blood into the fetus, and the waste material of the fetus being taken out and excreted via the mother's blood circulation was known. The Western reference to this came only in the 1600s, where the Italian physician Hieronymus Fabricius, in his book De Fornata Fieta on the formation of the fetus, first described circulation in the umbilical cord. So we knew it many years ago before the Western world described blood circulation in the fetus. Talking about deformities in the newborn, birth defects, in the Charaka Samhita, chapter 4, paragraph 30, the reasons for these deformities are outlined. So they say that the deformities are seen at 3 levels: in the seed, in a part of the seed, and in a portion of the part of the seed. To look at it analogous to current times, in modern day science and modern day genetics, it refers to the sperm and the ovum, the seeds. Sperm coming from the male, the ovum coming from the female. A part of the seed, the chromosomes that are present within the sperm, within the ovum, that is the DNA. So, a part of the seed could be defective, or a portion of the part of the seed could be defective, which means a gene, a particular small region on the chromosome could be defective. So 3000 years ago, our Vedacharyas, our Ayurvedacharyas knew about the sperm and the ovum. They knew that the union led to the formation of the embryo, the development of the embryo, and they also knew that the deformities in the child were a result of mutations in the DNA, either chromosomal abnormalities or other types of mutations in genes, and it was attributed to these and not to any supernatural powers. This was the science in Ayurveda 3000 years ago, and it was only in the 1950s that the field of molecular biology and genetics, as we see it today, with the structure of the DNA discovered by or proposed by Francis Crick and James Watson, came forth. And it was only after that that we began to study chromosomes and DNA at the molecular level that we look at today, and attributing genetic deformities or defects in development to genes. But we Indians knew it 3000 years ago. We shift focus now to another important branch of Ayurveda, which is pharmacology. Ayurvedic texts say that there is no plant present in this universe which does not have a medicinal property. According to the World Health Organization, there exist over 400 species of medicinal plants across the world. And more than 300 of these occur in India itself. That is because of our geographical diversity, because of the climatic diversity we see across our country. One of the most popular herbs that is mentioned in Ayurveda is ashwagandha or Withania somnifera. So in the Bhava Prakash Nigantu Guduchadi Ghanaha text in Shloka 179, this text is from the Vedic period. The advantages of using ashwagandha are mentioned. Its medicinal properties are mentioned. It said that it's an astringent, bitter medicine that removes doshas in vata and kapha. It cures leprosy. It helps to recover from general inflammation, the swelling of the body, a general debility, a feeling of ill health, and it energizes and enhances the sperm. Besides many other verses also talk about the detailed medicinal benefits of ashwagandha. The relation to current modern day time is that the Defense Research and Development Organization of the Ministry of Defense Government of India has used ashwagandha along with 14 other commonly used herbs into what is known as the Composite Indian Herbal Preparation 1. It's a mixture of 15 commonly used Indian herbs, which they are using to enhance, to boost the strength of our soldiers, of our Jawans. The ones especially who work in high altitude regions like the Siachen Glacier, extreme cold temperatures, those that work in high intensity stress areas, let's say like Kashmir or Arunachal Pradesh, which border China, which border our neighboring countries, and also those that are those soldiers that are used for regular maintenance of peacekeeping in the country, who have a low level stress, but throughout they face this stress throughout. So this CIHP1 formulation, through clinical trials, they have done detailed medical studies, documented, published this in peer-reviewed scientific journals. Through such studies, they have proven that this formulation is known to boost immunity, is known to increase the health and wellness, not just physical, but also mental of our Indian soldiers. So that is the current relevance of this herb and several other herbs like tulsi and curcumin, which or haldi, which form part of this mixture. Another polypill which is used to treat heart diseases even today, very actively used even today, has been mentioned in the Sushruta Samhita, and that is Arjuna tree extract, or Terminalia arjuna. That's the scientific name of the tree. So the extracts of this tree bark especially are known to help in joining disjointed bones, are known to destroy any illnesses that are caused due to phlegm formation, any kind of respiratory illnesses. It is known to cure heart disease, it's known to cure liver disorders like jaundice, and it is an antidote to several poisons and is also used to recover from any kind of injury. It's mentioned that those who drink the extract of the Arjuna tree bark along with ghee, milk, or molasses water, that's sweet water, live free of heart disease and fever and live with healthy blood in the body. It is no wonder then that Arjuna Rishta and other formulations made using different parts of the Arjuna tree, especially its bark, are used even today as potent medicines to cure and to manage cardiovascular diseases. Talking about the heart, we will discuss a little about cardiology that is mentioned in the Sushruta Samhita. The Sushruta Samhita outlines in chapter 14 the detailed structure of the heart. It describes the arteries, the blood vessels that supply blood to the heart, the veins that take away this blood or that bring back the blood to the um, heart. So the arteries carry oxygenated blood, the veins bring back deoxygenated blood for the purification, and it outlines this entire structure and functioning of the heart. Hridaya, the Sanskrit word for heart, is actually composed of three verbs. Harati, which means receives, Dadati, which means gives or propels, and yati, which means moves or circulates. So ru, da, ya, coming from these three verbs, and it talks about the functioning of the heart. What does the heart do? The heart receives blood, it pumps blood, and it circulates blood to all parts of the body. So the details of the structure and the functioning of the heart have been mentioned in the Sushruta Samhita. The Western references to blood circulation in the body and to cardiology in general come only in the 16th century AD, where a Spanish physician, Michael Servetus, described the circulation that occurs in the heart and the lungs. And it was only in 1628 AD that Sir William Harvey, whom we all have probably heard about, the English physician, described the circulation of blood as we know it today. But it has already been mentioned many years ago in the Sushruta Samhita. It's not just about these herbs, medicinal formulations, the structure of various organs of the body, but Ayurvedic texts also describe in detail various treatment strategies, the Shodhana and the Shamana strategies that we spoke about in the first lecture. So here's an example of a shloka from the Charaka Samhita that outlines Rasayana therapy, or the rejuvenation therapy, especially used in old age or for geriatrics. So it says that this Rasayana therapy promotes intelligence, it boosts memory, it boosts immunity, it increases the lifespan, it increases longevity. It strengthens the sense organs, stimulates the digestive fire that has been weakened with old age. It imparts good color to the skin, maintains skin health, overall health. It's an indication of overall health, and maintains a favorable state of vata. So similarly, different treatment strategies in detail have been outlined in these ancient texts along with their benefits and how to undergo those treatments. One of the most popular sections of Ayurvedic texts that we have heard of in current times are those on surgery. So outlined here are images and verses from the Sushruta Samhita, chapter 7, which highlight the instrumentation that is used for surgery. As you can see in the image, various types of swastika instruments or forceps are shown, where each of these have a slightly different shape and dimension, and hence a different function in surgical procedures. For example, you have the Simha Yantra, which is faced like the lion, the Vyagra Yantra like the tiger, the Dwipi Yantra leopard, the Mriga Yantra like the deer. And it's not just these animals, but there are also forceps shaped like the beaks of various birds that are seen. You can see like the uluka, or the pointed one like the owl, the grida like the beak of the vulture. You have the cock-faced or the Masa Yantra, the Sasaghata Yantra or the eagle-faced, and so many more. I would like to draw your attention to what has been listed here in English. It is not just these instruments which are larger in size, but the Sushruta Samhita also outlines instruments for removal of small foreign bodies from our body. For example, it talks about lentil size, dal sized nails, which are appended or attached to these forceps in order to extract these small thorns or small foreign bodies that have inserted into our body. Not just instruments, but detailed surgical procedures and post-surgical stitching that is done has been mentioned in the Sushruta Samhita. So in this shloka from chapter 25, this shloka deals with the kind of grass or the kind of fibers that can be used for stitching wounds. So it talks about those fibers obtained from the Ashmantaka grass or the Marwa creeper, which is shown here. The fibers of the Marwa creeper are so strong that even today they are used to make bowstrings. So these you can use silk, you can use linen, you can use sinew from the body, you can use hair, and various other plant fibers can be used to stitch wounds. It is not just what fiber can be used, but the Sushruta Samhita also outlines the various kinds of stitches that can be used. Horn-shaped stitches, straight knots, talks about various other stitches and also gives detailed illustrations to show how these stitches are made. So suturing as a science in itself was mentioned in great detail in the Sushruta Samhita. Talking about surgeries, which was the first surgery that we know about? The first surgery, be it in Western reference or Indian reference, is the trepanning of the skull. That is to bore a hole in the skull. And the European references mention this extensively. They talk about examples from various parts of Europe, from Africa, and from various ancient civilizations. But I would like to tell you that the tree panning operation and how it was done, what instruments are to be used, were mentioned in our ancient texts more than 3000 years ago by the ancient Indian surgeon Maharshi Jivakaha. He was the royal physician of King Bimbisara 500 BCE. It is documented in the text that he removed a centipede from the brain by performing this trepanning operation. He bored a hole in the skull and extracted this centipede. In current times, this is interpreted as removing a dog tapeworm. Tapeworms are these helminths that are found inside the human body that grow and develop there, and some of them can migrate to the brain. So this operation actually refers to the removal of this dog tapeworm from the brain. The most famous type of surgery, of course, that we know of from Ayurveda is rhinoplasty, or the restructuring of the nose, the plastic surgery of the nose. So what is documented apart from the Ayurvedic text in the British rule over India are the punishments imparted by Tipu Sultan. So not just Tipu Sultan, but the rulers of those days used to cut off the nose of the person as punishment of a thief or of a person who has known to have performed adultery or a person who has betrayed the state. The nose would be cut off as a sign of punishment. What the Britishers observed was that a Maratha Vaidya is Vaidyacharya is a doctor in Ayurveda, Ayurvedic doctor. He was seen to perform these rhinoplasty. So for these people who had their noses cut off, he would stitch back that nose using skin flap from the forehead. So from the forehead, he would use a part of the skin and use other mechanisms to stitch back the nose. And this is an actual photograph from those times, a painting from those times. And a detailed account of this was made by the British physicians of those days in the Madras Gazette, as also the detailed procedure was given in the Gentleman's Magazine way back in the 1790s. This procedure, this detailed procedure was then followed or reproduced by the British surgeon J.C. Carpew in Britain, and also the German surgeon Graf in Germany, where they followed the exactly same procedure and conducted successful rhinoplasties. So the question was, where did this Maratha Vaidya get this knowledge from? How did he know what to use, how to use, what to do to reconstruct these noses? And that is outlined in detail in the Sushruta Samhita, chapter 16, paragraphs 27 to 31. and this was in the 6th century BCE. So Shushruta says, I will tell you exactly the method of joining a disjointed nose. He outlines what material to use. So cut a skin from the forehead the size of a leaf. He tells you how to do this, how to reconstruct the nose. So use stalks from certain medicinal trees, use them to hold this skin flap over that part of the broken nose. How to do this? Lift up the nose, lift the nostrils, and then sprinkle what kind of Ayurvedic medicinal formulations, how and what to put on the operated region so that it does not become septic, it does not catch infection, and it promotes tissue growth and building back of that broken nose. Sushruta also outlines the diet that needs to be fed to this person during this post-surgery recovery phase. And he talks about the details of how this nose would grow back, or how that tissue would regenerate and hold in place those stalks that have been used to give back the nose complete with nostrils and complete functionality restored. So this detailed rhinoplasty has been mentioned in the Sushruta Samhita and has been redone, has been reperformed by surgeons in the Western world and known to be successful. So these are just some of the examples of the science that is present in Ayurveda in the Ayurvedic texts. And I hope that now you would be able to appreciate and not just disregard Ayurveda or any of the other traditional branches of medicine just like that. They are equal or even more so in importance and the scientific knowledge that they contain as compared to modern medicine. At this point, I would like you all to pause this video and answer this question based on our discussion till now. Match the items in column A with those in column B. Here are the answers. The blood circulation in the fetus was mentioned by Maharshi Bhela in his Bhela Samhita. Rhinoplasty, the details of which we just discussed, were given by Maharshi Sushruta in the Sushruta Samhita. The genetic basis for deformities in newborn, that these developmental deformities are caused by genes and DNA, mutations in the DNA and not by supernatural powers, was mentioned by Maharshi Charaka in the Charaka Samhita. And the trepanning of the skull, which is the first surgery known to humankind, was performed by Maharshi Jivaka way back in the Vedic times. Having realized the beautiful science that is there in the Ayurvedic text, the depth of it, we will now move on to compare this with modern medicine. Said about Ayurveda by Acharya Vagbhatta, ये तु संशोधनैः शुद्धा न तेषां पुनरुद्भवः. ये तु संशोधनैः शुद्धा. Ayurveda consists of proven principle. All research has been completed. Everything that is mentioned in the Ayurvedic texts has been extensively investigated and proven beyond doubt. Na tesham punarudbhavaha. No further changes are possible. Idam agama siddhatvat. Ayurveda has arrived already proven. As opposed to this science where everything has been proven, modern medicine is termed as Vidjnana or Vishesha Jnana. Modern medicine as we know it today changes as newer knowledge becomes available through progressive research. Older principles are let go of and newer paradigms are set in, as through modern day research using modern technology, new knowledge is acquired. However, with Ayurveda, everything has been already proven. What is to be known is known, and no further changes are now possible. This leads to a difference in the approach of Ayurveda and modern medicine towards health and disease. Ayurveda, as we have already seen, looks at the bigger picture. It is a holistic science where the Ayurveda Charyas, the practitioners of Ayurveda, are considered healers. They heal a patient completely, holistically, physical, mental, emotional, and spiritual healing. Over several generations, these healers who understood Ayurveda, who breathed Ayurveda, who lived Ayurveda, turned into medical practitioners. They understood the principles of Ayurveda and they applied them to treat people, to promote health and well-being. With time, Ayurveda and other traditional forms of medicine took a setback and modern medicine came into play. Modern medicine led to specializations. A medical doctor would be specialized to treat certain parts of the body, and in recent times there is super specialization where current medical doctors and researchers are looking at the molecular details. They're looking at molecular mechanisms of disease, of how to cure disease. So from a holistic approach from the bigger picture, we have come down to studying, to investigating molecular mechanisms, and this has led to a reductive approach or reductionist approach in modern medicine. What this has done is there has come in more compartmentalization. Functional boundaries have been created so that different super specializations exist in their own right. And this has led to a difficulty in understanding worldviews. This has led to a narrower but in-depth approach where we have lost touch with the bigger picture. So modern medicine is more reductionist in its approach, whereas Ayurveda is more holistic. Here are a few differences in the approaches between Ayurveda and modern medicine. One is holistic, one is reductionist, as we already saw. Ayurveda focuses on promoting health and preventing disease, whereas the focus of modern medicine is the disease itself and to cure the disease. Ayurveda promotes health and prevents disease by incorporating certain lifestyle and nutrition changes, supplemented with medication and treatment strategies. Whereas modern medication focuses more on the medicinal aspects and surgery to cure diseases. Ayurveda has a very sound rationale, as we saw in the first part of this lecture. It is based on experiential wisdom. The treatment strategies, the benefits of Ayurveda have been experienced by individuals and documented by the Ayurveda Acharyas. It has a very strong scientific basis, which comes from experience. Modern medicine, on the other hand, is more objective in nature. It is verifiable through various experimentation using modern scientific method, and it is based on evidence-based research. You carry out research, you make certain observations, and what you see is what you believe. Ayurveda, right from those ancient times, has always had a personalized approach to health and relief from disease. Ayurveda talks about the prakrutis, the vata, pitta, and kapha balance that is present uniquely in each individual. As opposed to this, modern medicine is more generalized. People with different constitutions are given the same medication despite a slight difference in the way they respond to this. This has been the approach of modern medicine. And lastly, to understand, to comprehend Ayurvedic texts, we need to have a complete mastery over Sanskrit language. In Sanskrit, different words can be interpreted differently. The same word has several different interpretations. So when an individual tries to interpret the Ayurvedic texts, different interpretations come forth, leading to a slight friction in the way these texts are interpreted. On the other hand, modern medical texts have all been written in English or translated into English. So there is one way to understand them. There is a single interpretation and it is understood uniformly across. So this is an advantage when it comes to understanding the fundamental principles of modern medicine over that of Ayurveda. In recent times, we have come to know that the manifestation of a disease or of health for that matter is more than the sum total of the individual interactions that are involved. It consists of the chemical characteristics of the biomolecules within our body. It is dependent on factors which are inherent, which are endogenous. It is dependent on factors which are external, like the external environment, the changing seasons. So exogenous factors or maybe pathogens that infect the body. And of course, the genetic makeup, the DNA, the genes that are present in each individual, and the alleles of those genes, the different forms of the same gene that are present in different individuals. So health and disease are a manifestation of a synergy of all these factors put together. And this results in the unique biochemical profile of an individual. This was known to Ayurveda more than 3000 years ago, and we have seen the details of this in the previous section. But this has been known to modern medicine only recently. Only in the past few decades have research been taken up where these endogenous, exogenous genetic factors and also the chemical characteristics of biomolecules have been studied and put together to understand health and disease. From a holistic approach in ancient India or in ancient civilizations to a reductionist approach in current day modern medicine, we are now going back to looking at the bigger picture. There is a resurgence of medical pluralism where different forms or alternative forms, traditional forms of medicine are gaining more and more acceptance. And this has led to breaking of the silos. Those functional boundaries that had been created by the reductionist approach, by the super specializations, are now being broken. We are more accepting of worldviews, of views from different systems of medicine, and we are going back to looking at a holistic picture, to looking back at the bigger picture. We are now overcoming these barriers which are philosophical or the fundamental premise of Ayurveda and modern medicine. We are overcoming barriers there. We are overcoming the linguistic barriers of Sanskrit versus English used in Ayurvedic texts versus modern medical texts. We are overcoming the historical, cultural and societal barriers that have shrouded the science that is present in Ayurveda over all these past generations. We are also now performing evidence-based research, carrying out many experiments to prove Ayurvedic principles using modern scientific method, using cutting-edge technology. And we are also making several changes at the policy level, especially in India, where we are more accepting of Ayurveda. The government of India, Ministry of AYUSH, has several schemes that we will talk about, and it is actively promoting not just Ayurveda, but other traditional forms of medicine which have existed in India from past several generations. And this course on Ayurveda as part of your Indian Knowledge Systems course is also an effort in that direction. The way ahead now is to keep an open mind and have a healthy mutual respect for all the branches of medicine, and not just medicine, but all our ancient wisdom and modern science. There has to be a healthy synergy. There has to be an integration of principles from over our past, present, and our future outlook so that we have an integrative approach in all respects and in the current context in respect to health and well-being. In this section of lecture 2, we have looked at the evidence of the precise science that existed in our Ayurvedic texts. We have also outlined the difference between the approaches of Ayurveda and modern medicine and looked at the way ahead. I request you to pause the video for a while and reflect on these questions. How is Ayurveda different from modern medicine? In its basic premise, in the way it is studied, in the language that is used. Please reflect on this. And also reflect on the different ways that can be adopted to get an integrative approach so as to have the best of both systems, ancient Ayurvedic principles and modern day scientific medicine, in order to achieve complete physical, mental, emotional, as also spiritual well-being. Do reflect on these points. Let us now look at some of the schemes proposed by the Government of India for the promotion of Ayurveda in the country and abroad. In the year 1995, the Government of India set up the Department of Indian System of Medicine and Homeopathy, which was later renamed to the Department of AYUSH in 2003. The aim of this department under Government of India was to promote the traditional forms of medicine. In the year 2014, this department was converted to a full-fledged ministry with the vision of reviving and propagating the traditional forms of medicine in India. It focused its attention on education and research in the area of Ayush, Yoga and Naturopathy, Unani, Siddha and Saug Rigpa, and Homeopathy. The Ministry of Ayush, as we know it today, has this website. It was accessed just 2 days ago, and this is a screenshot of the website. The Ministry of AYUSH focuses on 6 major areas. The primary one, of course, being healthcare in the AYUSH areas by setting up various healthcare services and centers across the country. In terms of education, the AYUSH Ministry focuses on imparting quality education, on maintaining the standards of education in AYUSH colleges. To maintain the quality and standard of the drugs and various formulations used in these traditional forms of medicine, the Ministry of AYUSH also has set up rules and regulations for good manufacturing processes, for the trade, for the import and export of these traditional medicines in India as well as abroad, and to foster partnerships and collaborations across institutes in the country and internationally as well. In the year 2000, the ministry set up the National Medicinal Plants Board, or the NMPB, whose mandate is to look into the conservation, the cultivation, the trade, and the export of medicinal plants that are grown in India. In terms of the 6th focus, that is research and development, the Ayush Ministry in 2014 set up 5 councils of research, one for each branch of Ayush. These councils have the mandate of carrying out evidence-based research to prove, using modern scientific methods, the knowledge that is present in our ancient books, in the ancient systems of medicine. These research councils also organize various research consortia, that is conferences, seminars, symposia, where various research groups across India and abroad who are working in these different areas come together and present and publish their research. So research and development has been promoted extensively by the Ministry of Ayush. Every year on the 10th of November, that is the foundation day of the Ministry of Ayush, it is celebrated as the National Ayurveda Day, with a whole lot of activities promoting not just Ayurveda but the other branches of Ayush as well, with public awareness programs, with various research conferences and many different activities conducted to commemorate that day. The central government has also set up the National Ayush Mission, or the NAM, and a screenshot of the Kerala government's NAM website has been posted on this slide. The NAM has the vision to ensure easy and affordable access to quality and cost-effective Ayush healthcare services across the country. More than 12,000 Ayush health centers have been set up under the Ayushman Bharat scheme to empower the public to self-care, to take care of their health, to reduce the disease burden and to reduce their out-of-pocket expenditure on illnesses. So what is done is the public is made aware of various nutritional and lifestyle changes that can be brought about at their end in order to promote their health and well-being. And also treatments are provided at these health and wellness centers. This is especially important for the various parts of India which are deprived, which are in remote localities, and which have a backward and tribal population, a majority of this proportion. So these National Ayush Mission health centers are a boon to people living in these areas. I will request you to pause this video and read through the vision and the objectives of the National Ayush Mission. Besides the NAM, the central government also has various Ayush schemes in place. For example, Ayurgyan, which promotes education and research in these Ayush disciplines, and it also provides training and capacity building programs for the same. Ayur Swasthya refers to funding programs for institutions, for organizations to upgrade their research facilities in the Ayush disciplines. There is the Champion Services Sector scheme, which encourages medical tourism, especially in the context of Ayurveda and yoga. So people from abroad can avail these facilities and come to India to be cured by these traditional systems of medicine. The other Ayush schemes are listed on this slide. There is promotion of international cooperation through various schemes, information, education, and communication about Ayush and Ayush schemes through various boards that have been set up by the Ayush Ministry. The Ayush Aushadhi Gunavatta Evam Utpadan Samvardhan Yojana that talks about various schemes and business aspects, rules and regulations, good manufacturing practices for Ayurvedic medicines and formulations. And of course, the conservation, development, and sustainable management of medicinal plants, as we discussed through the National Medicinal Plant Board activities. So these are the various schemes that the Ayush Ministry has. Not just this, but the central government also allocates a sizable portion of the budget for promotion of Ayush. So in 2024-25, uh, almost a 24% increase in budgetary allocation to the Ayush Ministry was observed. And this helps promote all these government schemes and also to maintain the affordable healthcare services through the NAM initiative of the Government of India. So in this section of the lecture, we looked at the formation of the Ayush Ministry, how it existed before 2014 and how it was converted to a full-fledged ministry in 2014, the 6 focus areas, the National Ayush Mission, the various Ayush schemes offered by the Government of India, and also the budgetary allocation to the Ayush Ministry. You may now pause the video and answer these questions. What does Ayush stand for? Enlist the various schemes of the Ministry of Ayush Government of India that promote traditional systems of medicines. What are the aims and objectives of the National Ayush Mission? We will conclude lecture 2 with an overview of evidence-based research that is currently being undertaken in the area of Ayurveda. We begin this section by talking about Ayurgenomics. It's a new field that integrates the concepts of Ayurveda with modern genetics research. What it does is it looks at the expression of various genes in the body and correlates them to the Vata, Pitta and Kapha Prakritis, to the individual constitution of a person. In addition, it just not looks at the expression of genes, but over and above that, it looks at the regulation of this expression. Which genes are upregulated, present in more quantity, which are downregulated, the effect of which is lessened. All of this is put together, the ancient knowledge using modern scientific methodology in the discipline of Ayurgenomics, and then this information is made use of to prescribe personalized dietary and lifestyle changes to promote health and well-being and to counter any illness that a person might be suffering from. This field of Ayurgenomics has 3 aspects. One is the discovery aspect, the other the validation of that discovery, and finally translating that validated information in terms of modern science, in terms of modern medicine. So let us quickly walk through this image that is shown here. In the discovery phase, what is done is various modern scientific technologies, what we term currently as omics technologies, are used to look at gene expression in an individual, and that is correlated to the Vata, Pitta, Kapha Prakritis, the 3 doshas, and also to the expression of those in terms of the physiological changes, the physique of a person. So that is termed as the phenotypic individuality. What is inherent? How does it express on the outside? This discovery phase then leads to the validation phase, where these individual differences in Prakriti are validated. You look at the Prakriti of various individuals by Nadi Pariksha and correlate it to the gene sequencing, to the genetic data obtained from modern scientific methods. This helps us to decipher the molecular mechanisms of action of Ayurvedic drugs of various traditional formulations. This also helps us to understand the health outcomes of how these drugs would act and how these drugs would affect individuals with varying prakritis. So the same drug, how does it act on an individual with a Vata predominance versus a Pitta versus a Kapha predominance. So these outcomes are validated. And this then translates into the field of pharmacogenomics, where the difference in genetic composition of an individual is used to determine how or to predict how that individual will respond to a particular dosage of a drug. So that is the discipline of pharmacogenomics, which then leads us to the integrative P4 medicine approach, modern medicine with a holistic approach, which predicts these diseases based on prakriti, based on genetic makeup, and then prescribes an individualized approach, a personalized approach to the treatment. This also helps us to repurpose drugs. A drug used for a particular disorder can now be used to treat other disorders. And it also helps us to identify different biomarkers and targets, drug targets within an individual's body, specific based on these three different prakritis. So Ayurgenomics helps us to come up with integrative P4 medicine, connecting ancient Ayurvedic principles to modern day techniques and modern medicine. So here is the approach to Ayurgenomics. We decide on a population that we want to study, both healthy and diseased. We isolate the DNA from these individuals. We identify which genes are present, and at the same time, we also do a Prakriti screening. So on the one hand, we are using modern techniques to sequence the DNA to identify what genes are present. And on the other, we are using Nadi Pariksha to identify the Prakriti, the Vata, Pitta, Kapha doshas of these individuals. We use modern technology or techniques like genomics, the study of the DNA, which genes are present, which of these are in the normal form, which of these are mutated or changed. We use epigenomics. How is this gene expression regulated? We study the RNA component of the individual, which is called transcriptomics, and we also study the genes present in the various microbes which are present inside our body. So we look at metagenomics. Through all these modern scientific approaches, we provide evidence-based research to identify the Vata, Pitta and Kapha prakritis. These studies are then validated in preclinical trials in animal models and using cell-based technologies. And finally, these results are validated in humans through human clinical trials. So this is the approach that Ayurgenomics takes towards drug discovery for personalized medication. One of the founders of this new discipline of Ayurgenomics is Dr. Mitali Mukherjee, who's the head of the Biosciences and Bioengineering Department at IIT Jodhpur. She holds a PhD from the prestigious Indian Institute of Science at Bangalore and has worked extensively in this area, connecting basic principles of Ayurveda to modern medicine. I urge you all to watch her YouTube talk on the subject. It is extremely insightful and enriching. Other than IIT Jodhpur, the CSIR IGIB Institute in New Delhi has a unit called the Trisutra unit that coordinates Ayurgenomics efforts throughout the country. It connects researchers, colleges, modern medicine colleges, Ayush colleges to conduct a collaborative research in the area of Ayurgenomics. I will request all of you to visit the website and find out more about this Government of India funded initiative. We now move on to another topic, which is network pharmacology. Modern medical approach to treating diseases has been that one drug targets one particular protein or molecule in the body, and that is cures a particular disease. So it's a one drug, one target, one disease approach. However, this poses many challenges to the safety, to the efficacy and sustainability of such a kind of treatment. In current times, when we are bombarded from all sides by various types of pathogens, just having one drug targeting one target and treating one disease does not seem as a feasible option. What modern science has done currently is to use the omics data from all the genomics, transcriptomics, proteomics, metagenomics analysis, and integrate it with polypharmacology. That is, poly meaning many and pharmacology referring to drugs and drug targets. So multi-target drug development. A single drug targets multiple proteins or biomolecules in the body. And this has given rise to the discipline of network pharmacology, which borrows heavily from the traditional medicines of Ayurveda and other traditional forms of medicine. The various formulations, the plant medicines, the animal-based products, all of these are multi-target. They are naturally multi-target, and current modern day medicine borrows from these to create this new field of network pharmacology. It affects both what is known as an interactome and what is the diseasome. So let's see what these terms mean. An interactome refers to protein-protein interactions in the body, the entire set of these protein-protein interactions. How do we build an interactome? We decide upon one protein that we want to study, and we look at what are the instances in scientific literature that we know that this protein reacts with other proteins. So our protein of interest is shown in orange. The other protein that it interacts with is shown in blue. We then build a network of interactions of all these other proteins with each other as well, including our protein of interest. So now we are building a protein-protein interaction network between all these proteins we identified in the first case. We then take this set of interactions and build upon it to identify many more such proteins and many more such interactions to generate a set of these interactions. And finally, we look at different individual proteins, like we looked at this orange one here. We look at different such proteins, build up their interactomes, and then put them all together to generate this diseasome, as it is called. So we correlate various protein-protein interactions with their disease. We do this for proteins present in a healthy individual. We build an interactome for protein-protein interactions in a diseased individual, and comparing the healthy to the diseased individual, we draw up a list of proteins and interactions that are typical of the pathology of the disease, that are present or can be correlated to a particular disease. So this is called interactome or interactome building. The other term is diseasome, where we look at the interactome for a particular disease. For example, we look at all the proteins involved and the interactions for mental retardation, for epilepsy, for various types of cancer, for diabetes, for obesity, for deafness, and all these diseases. And then we link them up together to identify the common players in these. And then we make use of polypharmacology, targeting more than one targets in order to treat these diseases. For example, if I target this set of proteins shown here and interactions shown here in purple color, I will be able to treat cataract, which is an ophthalmologic condition, and also gain some relief from mental retardation or mental disorders. If I target this set of proteins shown here in orange or in brown color, I will able to prevent the recurrence of not just one but several types of cancer, and so on. So this is how network pharmacology, that is network biology in combination with polypharmacology, borrowing heavily from traditional Indian Ayurvedic medications and formulations helps us to treat various diseases, to target multiple targets. This is the approach of network pharmacology. We identify the various bioactive compounds from Ayurvedic and other traditional forms of medication. We screen these compounds using modern scientific methods. We identify the potential targets, the proteins, the protein-protein interactions by performing a pathway analysis. Which are the metabolic pathways that are affected? Which are the diseases? So we build a disease zone. And together, using all this information that we have gained, we build the interactome. We construct the network. Network pharmacology approaches are valuable tools for evidence-based Ayurvedic research, for identifying the mechanisms of actions, to identify the indications of Ayurvedic formulations. On this slide, we look at interaction maps, the network pharmacology maps of two Ayurvedic components. One is called Nish Amalaki. It is an Ayurvedic medication and it shows you the interaction maps here. And the other one is for Haldi, for curcumin. And this is a diseasome. And what you see here are all the various proteins and interactions present in various diseases that Haldi can target. And to imagine that traditional Indian cooking uses a pinch of haldi every day in the various items, the food items that we make. So haldi provides a natural protection to all of this. Isn't this deep scientific wisdom hidden in our ancient traditions? Where do we get the information to create all these interaction maps? One of the databases that has its creation in India is the IMPACT database, which has been curated at the Indian Institute of Mathematical Sciences at Chennai. The IMPACT database, the name IMPACT stands for Indian Medicinal Plants, Phytochemistry and Therapeutics. This database is a compendium of information on all possible Indian medicinal plants and their usages, the chemicals that are present in these plants that are known to us through scientific research. The IMPACT database contains information from more than 100 ancient textbooks, from more than 7,000 scientific research publications in peer-reviewed journals, and other sources. It has information on more than 4,000 Indian medicinal plants, around 18,000 phytochemicals or plant chemicals that are present in these plants, and over 1,000 therapeutic uses for each of these chemicals. While using the IMPACT database, one can use any of the two approaches. One can either look for phytochemicals or these phytoactive reagents which are present in these plants, or one can search the database for therapeutic use. And both of these possibilities are there. In addition, the database also provides various chemi-informatic tools, modern day science technology, to analyze the data present in this database in terms of which molecule, what is its therapeutic use, can it be used as a drug, various parameters can be checked to look at the druggability of the targets as well as to look at the characteristics of drugs. I would urge you to visit this website and to look at the wealth of information on Indian medicinal plants that are used in Ayurveda regularly. We now move on to the last part of this talk, where we talk about P4 medicine as a means of evidence-based research. In one of the earlier sections, we spoke about how modern medicine is now going back from a reductionist approach to having a holistic approach, looking at the whole patient as such. This branch of medicine is called as P4 medicine, and it focuses on 4 Ps. What are these 4 Ps? It is predictive in nature. So it uses modern day genetic analysis tools like genomics, transcriptomics to analyze the genome of an individual and to look at the genetic makeup of that individual. This helps us to predict what are the diseases that the individual might be susceptible to. The second P stands for preventive. Having known this genetic makeup of the individual, we are able to predict the disorders and therefore to empower the individual to identify certain signs and symptoms of the diseases that they might be susceptible to at the very early stage when these diseases might be reversible. In this sense, it is preventive. Next, this is participatory. The individuals are informed about their genetic makeup, they are informed about the susceptibility to various diseases, and they can make informed choices about their health and healthcare services that they might want to avail. And in this manner, the P4 medicine is also participatory. The individual participates in their health promoting and recovery of health after disease, and therefore this entire process becomes much more effective. And finally, the fourth P stands for personalized, something which Ayurveda has been doing for the past 3000 odd years, but is only now being seen in modern medicine. Having all this information at hand, personalized treatment strategies using modern medication can be devised. Personalized dosing strategies, personalized medication, how frequently to take that medication can be devised for each individual based on their genetic makeup. So this is what P4 medicine stands for. Essentially, it is a synthesis of systems biology approaches that we discussed, the genomic, transcriptomic approaches, using digital technology, making use of bioinformatics, a lot of computational biology, and it is consumer-driven healthcare and social networks. So all this put together gives you the P4 medicine of current day, which is predictive, preventive, participatory, and personalized. P4 medicine also is important for pharmacogenomics. In fact, pharmacogenomics, the branch of pharmacology that looks at how an individual's genetic makeup leads to a difference in response that that individual shows to a drug. Different individuals are given the same drug, the same dosage form, the same frequency, but they show a difference in the drug response. For example, the same drug in the same dose, some individuals might show the expected positive normal response. Some individual might show an altered response, not exactly what is expected. Some might not show a response at all, might not respond to that treatment. And in some individuals, it might in fact have an adverse response. It might show some adverse effects. And in some rare individuals, because of their particular genetic makeup, it might even be toxic. The treatment might be fatal and the patients might die. All this is accounted for in P4 medicines because we are looking at individualized medicines, personalized dosage forms. So what we are essentially doing is looking at the right dose of the right drug for the right indication for the right patient at the right time. Something that is the basic premise of Ayurveda, just the terms used are different. We use Vata, Pitta, Kapha, Prakrutis to personalize the medications in Ayurveda. And what we use here is pharmacogenomics to ensure P4 medication for individuals. Let us look at P4 medication in using a case study. So we are going to look at an anti-leukemia drug. Leukemia is blood cancer. So we are going to look at a drug called 6-mercaptopurine or mercaptopurine in short, which has this particular structure, and it is a drug that is used to treat blood cancer. So now, based on an individual's makeup, most of the individuals in the population will be fast metabolizers for this drug. They will metabolize or break down the drug in the body very quickly, and that is why most population requires this drug to be consumed in large amounts and to be consumed frequently. That is what it demands, the fast metabolizers demand. But some people in the population are slow metabolizers. Based on their genetic makeup, they will take a longer time to metabolize the drug, and therefore giving the drug in the standard prescribed amounts might become toxic to them because the drug is in very large amounts, accumulates in large amounts in the body. And then we have that rare population which are extra slow or ultra slow metabolizers, to whom that standard dose of medication might prove fatal. They might even be killed because of it, because their body is unable to metabolize and detoxify it in the given time. This drug, 6-mercaptopurine, acts on a drug target, acts on an enzyme inside the body called thiopurine methyl transferase. Now, in the body or in the DNA of the fast metabolizers, DNA is present on as chromosomes, one chromosome coming from the father, one chromosome coming from the mother. So we have two copies of each gene present in our body. So in case of the fast metabolizers, both the copies of the gene are normal, or they are what is termed as wild type copies. In the case of slow metabolizers, one copy is normal or wild type, the other copy is mutated. In the case of ultra slow metabolizers, both the copies of this gene, which gives rise to this protein, thiopurine methyl transferase, both the genes are inactive, they are mutated, and therefore the protein itself is mutated, and that is why these individuals cannot metabolize this particular drug. Having known this genetic makeup, we can tailor the drug dosage to fast versus slow versus ultra slow metabolizers. So for fast metabolizers, we can prescribe a higher and more frequent dosage, whereas for an ultra slow metabolizer, we would prescribe lower amounts of the drug and also at lower frequencies. So just to represent this graphically, in a conventional dosing method, all these three types of populations, the wild type, which is fast metabolizer, the slow metabolizer, and the extra slow metabolizer, would be given the same drug dosage, which could be toxic to the slow and the ultra slow metabolizers. But now that we know of this genetic makeup of each type of population, the drug dosage can be individualized. It can be customized. So the wild type or the fast metabolizers get a particular dose which is higher than that compared to the slow versus the ultra slow metabolizers. The ultra slow metabolizers get a very, very small amount of that particular drug. And if you notice this shaded area and compare this particular green graph to the one above, you would note that even for this normal population who metabolizes the drug normally, we need not give such a high dose. Even half that amount is effective. Isn't this excellent for ensuring the safety when we take all these medications? So this is how pharmacogenomics and P4 medicines help us to customize, to personalize the drug in the modern medicine context. So far in our lecture, We have looked at evidence-based research that is undertaken in the discipline of Ayurveda through Ayurgenomics being conducted at various institutes throughout India. We looked at network pharmacology, interactome, diseasome, and the impact database that stores all the information required to create these network pharmacology maps or interactomes. And we ended this section by discussing P4 personalized medication, which is the holistic approach that modern medicine is moving towards today. Please pause the video now and answer these questions. Define and explain the terms Ayurgenomics, network pharmacology, and P4 medicine. To summarize lecture 2 on Ayurveda, we began by discussing specific examples from the ancient texts of Ayurveda, where we discussed the science that is mentioned there in terms of embryology, pharmacology, surgery, cardiology, and others. We looked at the difference in the basic premise of Ayurveda versus modern medicine. We looked at the differences. We also discussed how we need to bridge this divide, how we need to have an integrative approach where we have best of both worlds coming together. We discussed the Government of India initiatives of the Ministry of AYUSH in terms of propagation of the traditional systems of medicine in India and abroad. We looked at the various government schemes that are in place for this. And towards the end, we looked at Ayurveda in the post-genomic era, where we looked at how modern scientific tools and scientific methodology is being used to prove the fundamental principles of Ayurveda beautifully in terms of Ayurgenomics, network pharmacology, and P4 medicine. As we went through all these topics, we also looked at the way ahead for Ayurveda and for modern medicine, where we said we need to have an integrated approach. We have to keep an open mind where we take the principles from both these disciplines, in fact, from all the disciplines of medicine together, and have a holistic system which will benefit the health and well-being of all individuals. We have to keep an open mind. We have to be proud of our ancient systems of medicine and ancient knowledge, ancient wisdom, and we have to own it. It is ours and we have to be proud of it. These are some of the reference books that I referred to, two of the major ones from where my content has come. One is called Ayurveda Unraveled. It's a beautiful book written by allopathic doctors who have been researching Ayurveda for many, many years. And also a book titled The Pride of India, which talks about the scientific heritage of India. It has been published by Sanskrit Bharati in 2006. And all my examples from the ancient texts and their modern context has been taken from this book. I would also urge you to look at the online resources that I have shared through these two lectures. Some of them I will also be sharing in the study pack that will be shared with you. I especially urge you to visit the Ayush Ministry's website and to look at all the initiatives that the Government of India has been taking to promote our ancient Indian science. For those who are interested in the scientific aspects, here are a few review and research articles that you can refer to. These are at a slightly higher level, but I do urge you to skim through them in order to get the actual scientific data, the evidence-based research that I have been talking about. With this, I end my two-lecture series on Ayurveda and its journey from ancient to modern times. I hope that this series of lectures has been insightful and enriching for all of you, and that now collectively we will take the responsibility of furthering this ancient science which is there in our ancient texts, and also to be proud of this ancient wisdom of India. Thank you.